Today, FDA approvals in small cell lung cancer and of a biosimilar, a European approval in ovarian cancer, and Japanese approvals in acute myeloid leukemia, ovarian cancer, and NTRK-positive tumors. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has approved pembrolizumab for the treatment of patients with metastatic small cell lung cancer who have disease progression on or after platinum-based chemotherapy and at least one other prior line of treatment. The approval is based on pooled results from cohorts of the Phase 2 KEYNOTE-158 and Phase 1b KEYNOTE-028 trials. Data demonstrated that the PD-1 inhibitor elicited a 19% overall response rate in patients with SCLC who had disease progression on or after platinum-based chemotherapy and one or more other line of treatment. The ORR from pembrolizumab comprised a 2% complete response rate and a 17% partial response rate. Moreover, among the 16 responding patients, 94% had a duration of response of at least 6 months, 63% had a DOR of at least 12 months, and 56% of patients had a DOR lasting longer than 18 months. The accelerated approval for pembrolizumab is contingent on the results of a confirmatory trial. Pembrolizumab is also being explored in combination with etoposide and platinum-based chemotherapy in patients with newly diagnosed extensive-stage SCLC in the ongoing Phase 3 KEYNOTE-604 study. The FDA has approved the trastuzumab biosimilar ABP 980, known by the trade name Kanjinti, for the treatment of patients with HER2 overexpressing breast cancer, as well as HER2 overexpressing metastatic gastric or gastroesophageal junction adenocarcinoma, marking the 5th approval by the agency for a trastuzumab biosimilar. Results of the Phase 3 LILAC trial, which were presented at the 2018 ASCO annual meeting, showed that ABP 980 demonstrated equivalent pathologic complete response rates and similar safety findings to that of reference trastuzumab, based on central laboratory evaluation in patients with HER2-positive early breast cancer. There were no clinically meaningful differences between the two products. The approval follows a rejection from the FDA in May 2018 when the agency rejected the biologics license application that was submitted in July 2017. The European Commission approved ABP 980 also for the treatment of patients with HER2 overexpressing breast cancer, as well as HER2 overexpressing metastatic gastro or gastroesophageal junction adenocarcinoma in 2018. In ovarian cancer, the European Commission has approved olaparib as a single agent for the maintenance treatment of adult patients with advanced BRCA1/2 mutated germline and/or somatic high-grade epithelial ovarian, fallopian tube, or primary peritoneal cancer who are in complete or partial response following first-line platinum-based chemotherapy. The approval is based on results from the double-blind Phase 3 SOLO-1 study, in which maintenance olaparib led to a 70% reduction in the risk of disease progression or death versus placebo in patients with BRCA mutant advanced ovarian cancer after completing frontline platinum-based chemotherapy. The median PFS by independent central review was not reached in the olaparib arm versus 14.1 months in the placebo arm. The investigator assessed PFS in the olaparib arm was not reached versus 13.8 months in the placebo arm. The FDA approved olaparib for this indication in December 2018. Shortly after the European approval, Japan's Pharmaceuticals and Medical Devices Agency approved the PARP inhibitor as a maintenance treatment for patients with BRCA mutant ovarian cancer after undergoing first-line chemotherapy, also based on the SOLO-1 data. Olaparib is the sole PARP inhibitor that is approved for use in Japan. The Ministry of Health, Labor, and Welfare of Japan has approved quizartinib for the treatment of adult patients with relapsed/refractory FLT3-ITD-positive acute myeloid leukemia, as detected by an MHLW-approved assay. The approval of the second-generation FLT3 inhibitor is mostly based on the Phase 3 findings of the QuANTUM-R trial, as well as a Phase 2 study, which demonstrated that oral quizartinib showed a statistically significant improvement in overall survival compared with chemotherapy in this patient population. Results showed that quizartinib led to a 24% reduction in the risk of death versus chemotherapy. The median OS was 6.2 months in patients on the quizartinib arm, compared with 4.7 months in those who received salvage chemotherapy. In May 2019, the FDA's Oncologic Drugs Advisory Committee voted 8-3 against approving a new drug application for quizartinib for adult patients with relapsed/refractory FLT3-ITD-positive AML after conducting its own efficacy analysis. And this week, the FDA issued a complete response letter officially denying the new drug application for quizartinib. Entrectinib, known by the trade name Rozlytrek, received its first regulatory approval as Japan's Ministry of Health, Labor, and Welfare approved the drug for the treatment of adult and pediatric patients with NTRK fusion-positive advanced recurrent solid tumors. The approval is based on data from the Phase 2 Star Trek II, Phase 1 Star Trek I, and Phase 1 ALKA 372001 trials, as well as findings from the Phase 1/2 Star Trek NG pediatric study. Specifically, in Star Trek II, entrectinib achieved an objective response rate of 56.9% in patients with NTRK-positive solid tumors, and the median duration of response was 10.4 months. Among patients with central nervous system metastases, the intracranial ORR was 50%. Moreover, Japanese regulatory authorities are also in the process of reviewing a potential indication for entrectinib for the treatment of patients with ROS1 fusion-positive, locally advanced or metastatic non-small cell lung cancer. This week, we sat down with Dr. Kathleen Moore of Stevenson Cancer Center to discuss the role of Brachy mutations in advanced ovarian cancer.
Identification of germline mutations such as BRCA1 or 2 is basically essential in epithelial ovarian cancer. It's a must-do. Now, patients can decline testing, but all patients should be offered. So that's really the most important message, is there's no selection for who gets offered testing, and hopefully who's tested. Everyone should be tested. And why is that? Number one, if we identify patients who have a germline mutation in BRCA1 or 2, which is about 17 to 18% of high-grade serous and high-grade endometrioid, 17 to 18% depending on the population, it does give us prognostic information. So we do know from epidemiologic studies that women who have a BRCA mutation or BRCA-related ovarian cancer tend to have a longer survival, owing in large part to the increased sensitivity to chemotherapies, especially DNA-damaging chemotherapies that having a BRCA mutation brings. So the BRCA mutation, unfortunately, led to the development of the ovarian cancer, but it also can be kind of an Achilles heel for the cancer in terms of its response to chemotherapy. So it gives us information about prognosis. It gives us the opportunity to offer testing to first-degree relatives. So this is called cascade testing. So if you have a patient with a known BRCA germline mutation, then her brothers and sisters, so it's not a sex-linked mutation. Both men and women can be a carrier. It's a 50% pass-down rate with each generation. So all of their siblings should be offered testing. And if they test negative, then their children are not at risk. But if they test positive, then you have the opportunity when their children come of age to offer them testing as well. And if you identify subsequent mutation carriers before they get cancer, you can create what some would term previvors, where you can do things that aren't small. You can do risk-reducing surgeries to prevent the breast cancer, to prevent the ovarian cancer from developing in men. With BRCA2, prevent breast cancer. There's different surveillance for BRCA2 because there's risks with pancreas cancer, for example. So there's high-risk clinics that these patients can be referred to, and we try and prevent them from becoming our patients, which is really the ultimate goal. So cascade testing is critical, and we don't do a great job of it in this country. And the third reason to know is that it's predictive biomarker for response to DNA-damaging therapies such as carboplatin, but more importantly now, such as PARP inhibitors, and there are several other assets in the pipeline that may come to availability as well that these patients will derive benefit from. So if we identify these patients, we know that they will benefit from a PARP inhibitor, and they need a PARP inhibitor with frontline therapy. So knowledge of germline is incredibly important. There is about another, we think 7%, maybe it's a little more, but right now we think about 7% of patients who, if you test their tumor, they have a deleterious BRCA1 or 2 mutation as well. So they have no genetic predisposition and they're not at risk of passing on anything to children, but their tumor looks like it has a BRCA mutation. And the importance there is, again, the predictive biomarker of potential of that finding is that they also respond just as well to PARP inhibitor use as germline patients. And so they derive all of that benefit as well. So we want to make sure that we identify those patients so we don't deny them the opportunity to access a very effective therapy.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.